Good evening, everyone. Good evening, everyone, guys. Good evening, guys. Am I audible? Hello, everyone. Good evening, guys. I hope I am audible and visible to you all. How are you all, guys? Good evening, good evening, good evening, guys. Welcome back, welcome back. Hello, everyone. Good evening, good evening. So, how are you guys? So, we had a small gap yesterday, and we will continue today. And as you can see, the topic today, we are going to talk about Kingdom Animalia. So, a real topic which we are going to start today. Other topics were also real only, but they were the basics part. Now we are going to start with the animal kingdom, proper animal kingdom. So let's wait for 1 or 2 minutes. Other students can join as well. Then we can start. Sir, is this a chapter? Yes. It's a proper chapter we are going to talk about. Yeah, Atmika, yes, a PDF will be sent soon to you all. I remember I have to send the PDF. Namaste, namaste everyone. Good evening, guys. So let us start. Shall we start, guys? So I think last time we talked about the terminologies which are used in the zoology or as a whole in biology. Those terms are used. And now we start with the next topic, that is Kingdom Animalia. So once more, a last check, guys. Just give me a thumbs up that you are able to see me and hear me. I am audible and visible to you all. Just give me a thumbs up, guys. The screen is visible. Nothing is blurred. Yes, Yashwant, as plants have scientific name, animals also do have scientific name. All living organisms, in fact, we can have the scientific names. Sir, I think it's over for fun. Right. So when come goal 100 results? Yeah, that's a good question, but I also don't have the answer for it. Very soon, I think very soon it will be declared. So yesterday, why there was no class? Yeah, beta. Yesterday, I also don't know, beta, what is the actual reason. Might be some technical issues. But no problem, we can continue today. So let us start with Kingdom Animalia, guys. Yeah, paper discussion was also there at the same time. Yes, that might be the good reason that things were clashing, right? Yes, yes. Paper discussion was there. Chaitanya, no thunderstorm in Vijayawada. I think if you're from Vizag people are getting some thunderstorms right there. Here, no thunderstorm as of now. If suddenly it changes outside, I don't know. Okay, guys, so let us start with Kingdom Animalia. In your lower classes, in your 9th or 10th standard, you might have studied a little bit about Kingdom Animalia, Animal Kingdom chapter, in which you talk about the classification part of the animals, on what basis the animals are classified, and then we talk about the phylums. What are the different phylums present in Kingdom Animalia? However, there are roughly 36 phylums which are there in Kingdom Animalia, but we are not studying in that detail. For our curriculum, for the NEET curriculum, we are studying at most 10 phylums as per our syllabus. And again, a very important chapter, at least 2 to 3 questions. At least 2 to 3 questions every year in the NEET exam, it is expected. And very easily we can catch those marks, very easily catchable marks. Okay, little bit conceptual points are there, and majority it is depending upon your, by hearting skills, remembering it with some technique, some concept application, and then remembering the points and the examples related to those phylums. Okay, That's how we can understand. So let us start with kingdom Animalia, guys. Okay. So talking about kingdom Animalia, I have divided this topic into module wise. We will discuss module basis. Yes, mnemonics are the great help. We can use mnemonics as well. But first we will, understand. Actually, I'm not a great fan of mnemonics actually. I rather believe in understanding it very well. And once you've understood very well, then automatically there is a very less use of mnemonics. But yes, I will not deny mnemonics are also handy sometimes. So yes, a bit of conceptual application, understanding part, and then we can apply mnemonics also, especially for examples which we have to by heart. Okay, so let us talk about the module 1. What we will learn in this module today. So basically, quickly, we will go through characteristics of animals. What we will talk about, guys? We will talk about characteristics of animals. So can quickly you tell me few characteristics? Let's see. What are the characteristics of animals? What is only seen with respect to animals, not seen in plants? Come on. What is only seen in animals, not seen in plants? Very good. Movement. Movement can be seen in plants also, beta. Movement, root movement, stem movement towards the light. First point is locomotion, changing the position. Animals show locomotion. Very good. Thumbs up. Thumbs up for locomotion. Perfect. Next point, one more good point. Heterotrophic nutrition. Very good point. Someone wrote in the chat box, heterotrophic nutrition. Yes, animals are dependent on others for nutrition. We are not able to synthesize our own food. We wish we could synthesize our own food, but sorry, we are not plants. We are not able to do photosynthesis. Okay, after nutrition, after heterotrophic nutrition, another point, guys, someone wrote reproduction. See, reproduction is a basic feature seen in all the living organisms. So plants also show, bacteria also show, animals also show. So little bit more emphasis. Yes, Guru Sohail, absolutely right. Sexual reproduction. Sexual reproduction is a characteristic of animal. Mostly, majority of the animals, they show sexual reproduction. However, in some lower organisms, we may see asexual reproduction as well. Like hydra, you might be knowing hydra's budding. Sponges also fragmentation part. Planaria also regeneration part. You might be knowing little bit, right? Yes. Very good. Okay, now let's see what are the characteristics quickly, okay? So let's see what are the characteristics, guys. So kingdom Animalia, we'll talk about the introduction part and we will see what are the characteristics present. So animals exhibit wide diversity. So when we talk about animals, we see a lot of diversity in animals. Diversity with respect to what? What is with respect to diversity? What we can see in animals? Come on, let's see one by one, okay? What is the diversity we will talking about? So animals are what? So let's see one by one point. First of all, animals are multicellular. What are they, guys? They are multicellular. Second point, they are eukaryotic. Animals are multicellular. They are eukaryotic. Moving ahead, they lack cell wall. They lack what, guys? Cell wall. Very important feature because cell wall is lacking in most of the animal cell do not have any cell wall. Plants have cell wall, bacteria have cell wall, right? Fungi have cell wall. What doesn't have the cell wall is the animals. Very important point, right? Moving ahead. Next, what is another feature you told? Heterotrophic nutrition. What you said? Heterotrophic nutrition. Heterotrophic nutrition only seen in the animals. And very, very important, one more point. Animals have a well-developed nervous system. Animals have well-developed nervous system. They can respond to the external stimuli. They can respond to the external stimuli. And along with the developed nervous system, they also show specialized sense organs. Eyes, ear, nose, tongue. They have the specialized sense organ as well. Yes. Yeah, thinking. Yes, thinking definitely is coming through the nervous system part. And one more point, very important, as you were discussing in the chat box, many of you, Many of the animals are mostly sexually reproducing. Majority of the animals. When I speak general, I am removing some exception points. Like the lower invertebrates, lower non-chordates, I'm removing them. If you remove some of those animals, majority of the animal, the whole chunk of the animal kingdom is going to be what, guys? It is going to be sexually reproducing, right? So are we clear with this, guys? Yeah. See, adaptation, Yashwant, adaptation can be seen by plants also. Adaptation can be seen by plants also, animals also, right? So difference between the eukaryotic and prokaryotic, it is about the nucleus, beta. Eukaryotes have well-developed organized nucleus, not seen in the prokaryotes. Then the organelles part, organelles are not there in the prokaryotes. Organelles are present in your eukaryotes, right? Okay, so these are the basic characteristic of the animals, which will be seen in all animals, right? So these points, what you saw, these points, these are the basic characteristics of all the animals. Okay, moving ahead. Now, when I talk about, we are studying kingdom Animalia. In kingdom Animalia, we are going to talk about phylum. What we are going to talk about, guys? Phylum. Phylum is singular, plural it will become phyla. So roughly, if you talk about the phylums, there are roughly 31. Now I think the updated data is showing that there are roughly 36 phylums present in the animal kingdom. So very huge animal kingdom is there. A lot of phylums are there. Total 30 phylas. Okay. When I was making the presentation at that time, a few years back, I found out that 30 phylas have been developed or discovered so far. And from that 30 phylas, there are total 10 major phylums and 20 minor phylums. Okay. Right, guys? Phylum is a unit of classification, beta. Varshika, phylum is a unit of classification. So any animal, when you classify, see, when you study the classification topic, or if you have studied the binomial nomenclature, so if you see, whenever we classify any organism, we classify as per their kingdom, phylum, like that, class, order, family, genus, species. So these are the basic units of classification. Phylum is the second highest, you can say, the unit of the classification. After kingdom, there is phylum. Okay? And to be more appropriate, when you will actually study the topic of cell, the unit of life, you can study there that all the organisms are classified into 3 basic domains. Do you have any idea about domains? We know there are 5 kingdoms, right? We know there are 5 kingdoms: animal kingdom, plant kingdom, fungi hai, protista hai, and then you have the Monera kingdom, right? But above the kingdom, there are 3 domains. Okay, Mr. Carl Woese in 1991. Okay, Carl Woese in 1991 he classified the organism into 3 basic domains based on the ribosomal RNA. So this is little, extra part which we are going to talk about in your cell, the unit of life chapter. So you will understand that first there is a domain. After domain, then kingdom comes. And after kingdom, there is phylum. However, when you see the plant classification, in plant classification, you don't talk about phylum. In plant classification, you talk about after kingdom, the next highest unit of classification you will get is your division, right? Division you will get. Are you getting me, guys? Right? Are you getting me, guys? Give me a thumbs up if you are getting me. Okay. Right, guys? So total there are how many phylas? Total there are 30. Now they are updated to 36. Out of that, 10 are major phyla, 20 are minor. So here you can ask me, thank you for your thumbs up, guys. That's great. Okay. Or else I will be clueless. It's okay if some students are not getting it. Some of you might be, little lagging, okay, in understanding part, but that is okay. Absolutely okay. So you understand that there is kingdom Animalia. In kingdom Animalia, there is roughly more than 30 phylas. And out of the 30 phyla, 10 are major, 20 are minor. Now, what do you mean by major and minor phyla? Major phylum has a lot of species actually. A lot of species are there. So whenever an animal phylum has a lot of species, you consider it as a major phylum. When species number is very less, okay, then you will say it is a minor phylum. So our agenda is to study the major phylums. Our agenda is to study major phylums. And one minor phylum also we will discuss. We will study the major phylums and we will study one minor phylum as well. Okay. So, and this one minor phylum you might not have heard also, because it is a part of 11th standard NEET program. It is coming in the 11th standard NCRT. You might have heard of Porifera, you might have heard of Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Echinodermata. All these phylums you may have aware of, you might have heard of also. But one more minor phylum, Ctenophora. Ctenophora is the minor phylum which we are going to study. Okay. So moving ahead, guys. Right? Now, when I see any animal, if I see a dog, a cat, as you can see a dog, a cute little dog is there, a puppy, and a small, I think a rabbit or a hamster. It is rabbit, yeah. So it's a dog and a rabbit here, okay? So externally, if you look at the external morphology, they appear different, okay? Both are cute. But when you talk about the anatomical feature, here, internal structure, you will find a lot of similarity, right? Yes, guys? And if you see their nervous system, rabbit nervous system, same. Digestive system, almost same. Excretory system, almost same. Dog also have kidneys, rabbits will also have kidneys. Dog also has a liver, rabbit also has a liver. So when we talk about the animals, if you see majority of the animals, anatomical structure, external morphology might be different, external appearance might be different, but internally, a lot of anatomical features are showing a lot of common things, right? So on that basis, we are going to classify. What we are going to do? We are going to classify. So first of all, to classify animals, we have considered one basic point. We have classified the animals on a very important basis, that is notochord. Okay? So animals which have a hollow rod-like structure on their back or their dorsal side of the body, you call them as what? You call them as chordates. What do you call them as, guys? You call them as chordates. So look here, very important classification we are talking about. Animals are broadly classified on the basis of what? On the basis of a stiff rod called as notochord. Called as what, guys? Notochord. Called as notochord. Yes, Yashwant, that might be seen. That might be seen, but however, both are mammals, right? One is a herbivore, another is a carnivore. So very important basis of classification, notochord. So right now, what you have on your back, you call it as the vertebral column, which is holding your spinal cord. But when we go in the embryonic stage, the same vertebral column was in a less developed state, and that was called as a notochord. That was called as a what, guys? Notochord. So here we have a very simple basis of classification. All the animals which are there, we are classifying them on the basis of notochord. So animals which show presence of notochord, animals which show what, guys? Animals which show presence of notochord, you will call them as what? You will call them as chordates. You will call them as chordates. Animals, however, which don't show the notochord, they are called as the non-chordates. So we have two groups here, okay? Non-chordates and another one is chordate. So can you tell me what are you guys? Are you chordate or are you non-chordate? Are you a chordate or are you a non-chordate, guys? Come on, quickly. Basically, it is the first basis of classification, a very general basis of classification point here. Right? Yes, we all are chordates. We all are chordates. Very good. And talking about insects, talking about the insecta, which is coming from phylum Arthropoda, They don't show notochord-like structure. So they cannot be chordates, and they are called as non-chordates. Am I clear? So first basis of classification. Okay, moving ahead. Next point. Now let's see what are the major criteria for animal classification. Now we are going to talk about what, guys? One important basis of classification we saw. One important basis of classification was the notochord. Just remember that. Animals are divided into two parts. One is your non-chordate group, another is chordate group. Now we are going to see all the other criterial points on which the animal is going to get classified. First, habitat. First point is habitat. Do you think all animals live in a same habitat or they have a different habitat? Tell me. Come on, tell me, guys. Animals live in same habitat or different habitat? Come on. Tell me in the chat box. Same or different habitat, guys? Can I bring a penguin in Vijayawada? Will it survive? Can I bring this penguin to Vijayawada? Will it survive? No, absolutely not. Why? Because penguin's body is adapted. Penguin's metabolism is adapted to low temperature conditions. If you bring them in this condition, they will die. One day they will not survive. Correct? So basically, different animals survive in different habitats. Okay? Lion. Yeah, lion can survive. Lion is pretty much okay with the dry and humid, but lion will not survive us, na? We people first will kill them only. So next point, guys, come on. So these are the first two criteria which we did. First is habitat. Different organism survive in different habitat. So that is the first criteria. So when we study different phylum, like fishes only, if I talk about fishes, they live in marine environment also. They live in fresh water also. Can I bring a marine water fish and keep it in fresh water? Will it survive in those conditions? Or will I bring a freshwater fish like a rohu fish, rohu or katla, rohu fish, katla fish, or this, I think Apollo is a freshwater fish, what we get here locally. If you put in the marine water, what will happen? Will it survive? It will not. So the habitat is water only. Habitat is water, but the water has difference. Salt level difference is there, right? So freshwater fishes will not survive in marine. Marine will not survive in fresh. Right? Very few examples are there, however. Very few examples are there which can survive in both conditions. Like your salmon fish. Salmon fish mostly lives in your, marine water throughout the life. But for reproduction, it will migrate to the fresh water. So it can survive both the salinity level. You understand salinity level? Marine is more saline water, fresh water is less saline. So you can see some animals have good adaptation. Now here the adaptation point is coming slowly, right? Crocodile. Crocodile is very dangerous. It can survive marine also and fresh water also. So see, it has in both the habitats you can find. Yes, absolutely. Next, come on guys, done. So I hope you got a little idea. So when I talk about porifera, they are found in marine water as well as fresh water as well. Coelentrates, your jellyfish. Your jellyfish is mostly found in marine water, but the hydra, which is coming in coelentrates, that is found in fresh water. Right? Now, will you find starfish near your rivers? Tell me guys, have you seen starfish? It's not a fish though. Starfish is not a fish, water fish. So, have you seen a starfish nearby a river? No. You will not get starfish nearby river. Why? Because starfish is an animal which requires a marine habitat. So habitat is again very important, right? So every animal when we talk about, we will discuss their habitat also. Some are in marine water, some are in fresh water, some are found in both the waters. right? Some fishes are found in the estuaries. Have you heard the word estuaries? Are you getting me what I'm saying? Estuaries. Can you tell me estuaries? What is the meaning of estuaries? It's a habitat. What is the meaning of estuary? Yeah. Estuary, where I think your river water is, meeting the sea water. River water meeting the sea water, okay, and a little bit of backwater kind of thing is created there. And the salinity is very different, not that highly saline as marine water, and not that sweet as your river water, as your fresh water. So it gives a little different kind of salinity, okay. There you will find some other kind of fishes only. So there the fishes will be little different. Are you getting me, guys? So different habitat. Second, chalo. Habitat is done. So I hope I have given you one idea that animals are classified on their basis of habitats also. Different animals have different habitats. Okay? Then level of organization. Very, very important basis of classification. A lot of questions have been asked on the level of organization points as well. Basically, when I talk about organisms, the higher organism which you are looking at, mammals, amphibians, reptiles, birds, these animals, when you see these higher animals, their body shows organ system level of organization. What it shows? Organ system level of organization. Like there are multiple organs working in a coordinated manner, and that is making an organ system. Like when I talk about digestive system, a lot of things comes in your mind, right? Liver is there, pancreas is there, stomach is there, large intestine, small intestine, all these organs combine makes an organ system. Similarly, when I talk about the excretory system, you'll talk about the two kidneys, the ureter, single urinary bladder, urethra. They are making our excretory system, right? So when multiple organs are working in a coordinated manner, you will say it is organ system. So higher organisms, they show organ system level of organization. But when I go to very lowest of the organism, very lowest of the animal actually. So if I go to porifera, if I go in porifera best example, sponges. In sponges, if you try to find any organ system, you will not get it. In sponges, if you try to find any single organ as well as, you will not get it. In sponges, if you try to find any tissues also, you will not get it. But if you find some cells, you will find a lot of cells in sponge. So what I'm trying to say here is that sponges, the very basic of the animals, probably I can say in the evolution, they were the first animals, multicellular animals to be formed. And their body shows loose aggregation. They are aggregation, collection of loose cells. And that is the first level of organization, that how the body is organized of the animal. So it is cellular organization. See, beta, it is evolution. You need to understand this is evolution. So first when animals were formed, they were actually multicellular level, only some cells were getting collected and the organism's body was formed like sponges. But if you go one step ahead, after porifera, the next phylum you are very well aware of, after porifera, the next phylum coelentrates. Now, evolution always makes the thing best. Evolution always try to make the thing best. So what happened? Cellular organization. The cells combined to become what? Tissues. And from cellular organization in coelentrates, we got to see the tissue organization. What we got to see, guys? Tissue organization. So little bit more better. Work can be done better. Different tissue will perform different work. If you see a sponge body, porifera if you see, in porifera body you will see the cells doing all the functions. A single cell of a porifera will do respiration also, digestion also, excretion also, Every function it will do. But when you see in your coelentrates, in your jellyfish, different tissues are there. Different tissue will perform different functions, and you know very well, group of cells, yes, doing similar kind of functions. So different tissues were formed in your coelentrates. So little better, evolution, little better. Then after your coelentrates, we talk about the platyhelminthes. Okay, there is one more minor phylum in between, but that is also tissue level organization. So I'm jumping to platyhelminthes. Flatworms. If you see the flatworms, the tapeworms, the liver fluke, the planaria, okay, in them if you try to find, you will get some organs. What you will get? Some organs. And then we say, okay, evolution went further better, and in platyhelminthes and the flatworms we got the organ level of organization. What we got? Organ level of organization. Then we went to Aschelminthes, your roundworms. In roundworms, now if you see, like in the Ascaris, the filarial worm, from Ascaris onward, if you see, different organs are developed and they are working in a coordinated manner, and we got the organ system level of organization. So if you check in the animal kingdom, starting from Aschelminthes onwards till the whole your chordata, everyone is showing your what? Everyone is showing your organ system level of organization. Okay? Come on. Now let's move ahead. After level of organization, so see, I have given just a briefing, guys. Here we will discuss every classification criteria in detail. So first is habitat, second is level of organization. Okay, then we will talk about, after level of organization, there will be next point as symmetry. What will be next point? Symmetry. Okay, slide has went little bit behind, so we will get it back. Habitat ho gaya, level of organization ho gaya, then we will talk about symmetry. Okay, what is the symmetry of the animal? Is it divisible into two equal halves or not? How many planes are there that it is dividing the animals into two halves? Like if you see sponges, most of the sponges have a very irregular structure. You cannot divide them into two equal halves or mirror images through any planes. However, few exceptions are there in sponges, but majorly you will say sponges are asymmetrical. Then we have your coelentrates, the ctenophora, jellyfish and all. In their body, like if you take a jellyfish, a round body jellyfish, right? You cut in this plane, any plane which is passing through central axis, you will get two equal halves. Like you take a cake, you cut from this axis, you cut from the sagittal axis, frontal axis, any axis you cut, passing through central axis, it will be giving you two equal halves. That is a radial symmetry. That is which symmetry, guys? That is a radial symmetry. But then evolution occurred, a better symmetry started seen in the animals, and that is called as your bilateral symmetry. In bilateral symmetry, the body is divisible in two equal halves only through a single plane. Now, why symmetry is important? The evolutionary aspect is that getting the bilateral symmetry led to the formation of head and tail. So the animals which show bilateral symmetry, you can clearly see their head and tail area, starting from the platyhelminthes. So if you see the platyhelminthes, Aschelminthes, Annelida, Arthropoda, mollusca, there are some exceptions like snails, due to torsion, twisting of their shells, they are little asymmetrical. Then Echinodermata, the starfish, you can see the 5 arms, they will not show bilateral symmetry, they will show radial symmetry. Okay, and in the larva we will see the bilateral symmetry, that we will discuss later. Okay, so 3 types of symmetry we are going to discuss. After the symmetry point, what is next? So 3 criteria till now. After symmetry, what, guys? So base is body cavity. Next is what? Body cavity. Next basis is body cavity. Presence of body cavity. Can anyone tell me what is the body cavity also called as? The body cavity is also called as the coelom. Is also called as what, guys? The coelom part. Presence or absence of coelom. Many animals initially, the sponges, the jellyfish, coelentrates, they lack the body cavity. Okay, the body cavity coelom is not there. So we call them as acoelomates. Without coelom, acoelomate. Remember the word a prefix? Without it. Then animals evolution realized, okay, we need to make some body cavity to stuff the organs inside the organism. So we will make some cavity, okay. And then we got the cavity, okay. But initially the cavity was scattered in some pouches. So irregular small pouches were there. Then we call it as pseudocoelom, false coelom, like in Ascalmenthes. And then later, proper coelom was seen in the animals from the Annelida phylum. We call them as coelomates. Okay, guys. So this criteria is very important. Coelom criteria. On basis of this, we will classify the organism in three types: acoelomates, pseudocoelomates, and coelomates. Okay. Yes, beta. Yeah, Sohail. See, I am telling very fast because this is just a general point. When actual criteria comes, we will discuss this in detail. So no worries. All the criteria will be discussed in detail. Okay? Okay? Yes, Raghuram. I will send the PPT. No worries, no worries. After body cavity, which is the next criteria? The next criteria is germ layers. What is the next criteria? The next criteria after the body cavity is germ layers. Okay. Now, what is the meaning of germ layer? What does germ layer means? Germ layer is also known as the embryonic layers. Which layers? Embryonic layers. So basically, after fertilization, in animals, after fertilization, you get the zygote. What do you get, the guys? You get the zygote. Now, zygote is a single cell. Zygote is a single cell. From that single cell, you have to make an animal body which contains trillions and trillions of cells, right? So what we need to do? The zygote basically goes under division. It makes 3 layers of cells. What it makes? 3 layers of cells, which are called as the embryonic layer. So after zygote, we will get the embryo. Now in embryo, we will see the 3 different layers of cells, embryonic cells. And now those layers will be called as what? Those layers are differentiated. Those layers are differentiated into ectoderm, endoderm, and mesoderm. These are the 3 germ layers will be formed. Sir cannot remember all this classification. Neely, I am just telling the initial criteria words. You just note down the criterion words. Like first was habitat, second was level of organization, right? Body cavity, germ layers, first one notochord also. Just note down, okay? And if possible, I will share the presentation also. You can go through the presentation also later and make notes if you're not finding time to write it. Okay, next. So basically there are 3 germ layers: ectoderm, endoderm, and mesoderm. Ectoderm, endoderm, mesoderm. These are basically our 3 engineers. What are these? These 3 are basically our engineers. Ectoderm will make some tissue that will give rise to some organ. Endoderm will make some tissue, it will give rise to some other organ. Mesoderm will make some tissue and will give rise to some other organ. And like this, all the organ systems will be made by all these germ layers, right? So these are the engineers of your organ and organ systems. Who? Ectoderm, endoderm, mesoderm. They are also called as embryonic layer, germ layers. Now, sir, why we are discussing germ layers as a criteria of classification? Now, the thing is that Not every animal will have all these three germ layers. Not every animal will have all these three germ layers. Initially, the initial organism which have evolved, porifera, coelentrates, they mostly don't have these germ layers. Okay? They have only two. Sorry guys. Porifera doesn't have the germ layers. Coelentrates have only two germ layers. Coelentrates have only two germ layers. Yes, Sanday, I am talking about the criteria. Okay, so what I said that for any animal to be formed, we need fertilization of the gametes. Gametes will give rise to zygote. Zygote will differentiate into embryo, and embryo will make these germ layers. Embryo will make these germ layers ectoderm, endoderm, mesoderm. And this ecto, endo, mesoderm, which are known as embryonic layer or germ layers, they will be responsible for your organ and organ system formation. Now comes the criteria. What is the criteria we are talking about, sir? The criteria is that some animals don't have these germ layers. Like porifera, they don't have it. Okay, now here there is a lot of controversy regarding porifera. Some reference books say it is having it, some they say don't have it. But NCERT has not mentioned it, that porifera having germ layer or doesn't have. In the old reference books, you will find porifera shows two germ layers. Now the criteria I am talking about. Now if the animal is having two germ layers, see this. If the animal is having two germ layers, you will call it as diploblastic. If animal has how many germ layers? Two germ layers. If the animal is having two germ layers, ectoderm and endoderm, mesoderm, ledu. Okay. And if there are three germ layers, then it is called as triploblastic. So some of the animals are diploblastic. The initial ones, like your coelentrates and ctenophora, jellyfish and ctenophora, they are diploblastic, less developed. They only have two germ layers, ecto, endo. Whereas from your platyhelminthes onwards, from your flatworms onwards, from flatworms, that is platyhelminthes, till your chordates, till all of your chordates, they will have how many germ layers? They will have the three germ layers. Okay. So again, this is a very short description, guys. Again, please understand, very short description. The germ layers will be again taught to you in detail. Okay, in the further classes, in the gift classes only. But today I'm just listing out the criteria of classification. So moving ahead, after body cavity, germ layers. Okay, moving ahead, come on. Now after germ layers, which is the next criteria? After germ layers, what is the next criteria, guys? Then there is mode of locomotion. Very basic criterion. Germ layer is an embryonic layer, beta. Chaitra, it is a embryonic layer made up of embryonic cells. Okay. Next is mode of locomotion. Some animals are able to run. Some animals are able to repair and crawl. Some animals are able to fly. Some animals are able to swim. Okay. So different modes of locomotion. So while describing the different phylum and the animals, we will also talk about their mode of locomotion as well. Right? Clear? Okay. Moving ahead. Now, next criteria. Okay. So some animals can fly, some animals can run, some can crawl. So let's list down all the criteria. Come on. Let's summarize. Let's summarize what criteria we have talked about. Okay. Chalo. Let's summarize. Arey, locomotion. Sai, come on. Very basic, na? Changing of position from one location to another location. Next criteria, respiration. Oh, respiration also is a criteria. Do you think all animals respire the same way? Do you think all animals respire the same way? Or different animals have different respiration? No. Tell me, frog. Frog can respire via gills also. Frog can respire, breathe via skin also. They can also respire via lungs also. Can you respire via your skin? Or you only respire via your lungs? You only respire via lungs. Okay? So different animals will have different respiration structure. Can you tell me how earthworm respires? Yes, very good. Cutaneous respiration. Respiration by skin. Very good. Earthworm, how does it respires? Come on. Earthworm has a very moist skin. So earthworm is moist skin. Sponges, jellyfish, ctenophora, flatworms, they respire through their body surface. They respire through what? Body surface. So different animals will show different respiratory structure. Like in insect, there is a special tube for respiration or the gaseous exchange, the tracheal tubes. Exactly, Sheikh Mastan, correct. Tracheal respiration is seen in insects. So this is also one criteria. This is also one criteria. So every phylum when we discuss, we will see different mode of respiration. Okay? Next, don't write aerobic, anaerobic beta. All the animals are aerobically respiring. Majority of them are aerobically respiring. All of them require oxygen. Okay? Next, after respiration, what we can add? Excretion also. Very good. Like we remove our nitrogenous waste, urea, from where? It is removed in the urine via the kidneys. Kidneys are helping to remove them from the blood. What about the nitrogenous waste of fishes? How do fishes will remove their waste? Sir, how snake breathes? Snake is through lungs. How fishes will excrete, guys? Come on, tell me. How fishes will excrete? The nitrogenous waste of the fishes? Yes, very good. Fishes excrete the ammonium ions. The ammonia converted to ammonium ions through their gills. So they are using a different excretory structure. What about earthworm? What about earthworm, guys? Have you studied earthworm excretory structure? Nephridia. Earthworm shows a special coiled structure like your kidney has nephrons. Your kidney has a nephron structural and functional unit for the excretion of the waste. Similarly, earthworm has a nephridia. So here we are going to talk about different excretory structure in different phylum. So that is also a basis of classification. And similarly, guys, see, you can also say that excretory point, one more point is there in excretion. Some animals who are in water, majorly the aquatic animals, majorly they excrete ammonia as their nitrogenous waste. So they are ammonotelic organisms. Some animals like us, like mammals excrete our nitrogenous waste in the form of urea. So we become ureotelic. We become ureotelic because our maximum waste is urea. And if you notice birds, reptiles, snake, lizards, and birds, insects, they release their nitrogenous waste in the form of uric acid. Very good. So many students are writing uricotelic. So animals who excrete waste, uric acid, uricotelic. So see again, a small basis of classification we quickly did. Which kind of waste is released? What kind of structure is used? So again, this is a very important criteria. How different animals are excreting with different structures and different nitrogenous waste. Okay? Next, after excretion, guys, next, reproduction. After excretion, the next criteria will be reproduction. Lower animals like sponges, coelentrates, hydra, jellyfish, they are able to show asexual reproduction. Flatworms, they are also able to show asexual reproduction. Sponges show fragmentation. A small fragment of the sponge breaks off, goes in the water, and a new sponge is formed. That is fragmentation plus regeneration. In hydra, you may have seen external budding. A small hydra attached to a parent plant falling off, a new hydra is formed, right? So budding is there. See, lower animals will show asexual reproduction. But as you go above, as you go in Escalamenthes, Annelida, Arthropoda, Mollusca, Echinodermates, then chordates, majorly you will see only and only sexual reproduction. So lower one asexual, higher one majorly sexual, and then there is some differences as well. Majorly animals are unisexual. Majorly animals are unisexual, meaning dioecious, separate sexes, male, female sex are separate. But some animals like earthworms, flatworms, porifera, sponges, they are bisexual. They are bisexual, meaning monoecious, having both the sexes on the same body. So that part also we are going to study in reproduction. So two important parts I told you in reproduction. We'll talk about asexual, sexual reproduction. Then we will talk about which animals have separate sexes, unisexual or dioecious. Fragmentation example, sponges beta. Okay. And few animals like earthworm, flatworm, sponges, they are bisexual. Okay guys. So don't worry. Don't worry guys that I am giving a lot of examples you are not able to write. Just listen. All these things we are going to understand in detail. I'm just giving some examples so that you can connect the dots. Okay? Moving ahead. After reproduction, the next criteria. Wow. One more point came. Can human give eggs? Shelled eggs. Can human give shelled eggs? Tell me guys. No, humans cannot give shelled eggs. So one more point with reproduction, see, some animals are oviparous. The animals who lay eggs like birds, reptiles, majority of the reptiles, birds, who give eggs. And in eggs also, there are shelled eggs, without shelled eggs, cladoic, non-cladoic and all. Shelled and non-shelled, like amphibian eggs, frog eggs. Frog eggs matlab frog eggs doesn't have shell. But when you see the reptile, snake, lizard, or when you talk about bird eggs, they are having the shell. But whichever animal gives eggs, they are oviparous. Can you tell me the word which gives birth to young ones? In the chat box, come on. The one who gives birth to young ones, come on, yes, viviparous. Absolutely right. And mammals are viviparous, giving birth to young ones. Absolutely right. So see, so many points are there in reproduction. Asexual hai, sexual hai, unisexual hai, bisexual hai, oviparous hai, viviparous hai. And then one more point we can add. Larval stage. Which stage? Larva stage. Larva present, larva not present. Do humans have larval stage, do you think? Do you think larval stage is there for humans, birds? Do birds and humans have larval stage? No. But yes, when I talk about butterfly, when I talk about insects, mosquitoes, when I talk about the amphibian frog, tadpole, larva. So what you say? The larval stage is seen, yes. So animals which have larval stage, development is indirect. The development of the embryo is indirect. There is a larval stage and then it will become adult. But in some animals like humans, birds, reptiles, there is no larval stage. Okay? That is called as a direct development. One more point I gave you, direct, indirect development. Okay? Now, after reproduction, the next criteria, I think the criteria points are over now. Okay. Whew. So lot of criteria, yes. Lot of criteria you have seen. So let's see if we can compile all those criteria in one place. What criteria we just saw today. And in the next class, we will talk about every criteria one by one. Okay. So let us see what are the different criteria. Come on guys. Chalo, module 2. Criteria for classification. Okay. Okay, we are directly starting from the level of organization. So guys, you tell me what criterias we studied today. Come on. What is the criteria of basis of classification? Come on, guys. Chalo. Criteria. How many criteria did you saw for the basis of classification? Okay. Chalo. Quickly. First. What was first? Tell me. You discuss habitat. Different places, different habitat. Second, you talked about level of organization. 4 level of organization: cellular, tissue, organ level, organ system level. Come on. After organ, come on. After organ level of after level of organization, come on guys, come on. Symmetry, very good. Then it was symmetry. After symmetry, come on guys. After symmetry, what? After symmetry, yes, I know. Time is up. Body cavity, super. Body cavity. On the basis of body cavity, we classified it as having no body cavity, acoelomate, false body cavity, pseudocoelomates, true body cavity, coelomates or eucoelom. After body cavity, guys, come on. One more player said that notochord. Very, very, very important point. Notochord. On the basis of notochord, we classified as non-chordates and chordates. Come on. Five criteria are done. 6th criteria, guys. Come on. What is the 6th criteria? Come on. After notochord, what is there? After notochord, very good. Germ layers. As per germ layers, we have classified the animals into 2 groups. Having 2 germ layer, diploblastic. Having 3 germ layers, triploblastic. Come on. After germ layers, guys. Come on. After germ layers, quickly. जर्म लेयर के बाद क्या क्या बोलोगे? जल्दी बोलो. रेस्पिरेशन बोल दो. Different animals show different respiratory organs. Then different excretory organs are there. Excretion also. Then I can write reproduction also. Okay. Come on. Any criteria? You can find something else criteria, guys. Locomotion also. Yes. Different mode of locomotion is there. 9th के बाद 10th आना चाहिए ना भाई. ये locomotion हो गया. और do you know some have open circulatory system, some have closed circulatory system? Some animals have open circulatory, some animals have closed circulatory. In closed circulatory, the blood is roaming within the blood vessels, artery, vein, capillary. And in the lower organism like the arthropoda and mollusca, you will see the blood is present in the body cavity. Blood is bathing all the organs. The organs are getting bathed by the blood. So that is circulation. So I can add circulation point also here, guys. Okay. So many criteria are there, guys. Okay. One criteria we missed, which we'll discuss in the next class, that will be segmentation. That will be what, guys? Segmentation criteria. We will go ahead and discuss in the next class. Okay. About segmentation also we will talk about. So these are the criterias, and all these criterias, yeah, in segmentation, metamerism will come, beta. In segmentation, metamerism will come, Shamika. Okay, so that's it, guys. Thank you so much. That's all for this class, guys. Bye-bye, bye-bye, bye-bye. That's it. Can you teach classes reopen again, sir? Yes, beta. This chapter will be again revised. We will start from again. I hope you understood something, guys. So thank you all. See you in the next class. Right? Bye-bye. Take care, guys. Thank you so much.